Welcome to our tutorial on running the MetaCluster software. So to get started, we go to the GitHub page where the R package is located, and basically we've compiled a small tutorial here as well, where you basically can follow all the instructions in order to get the system running. And maybe first run it on the demo datasets, and then you can also use these demo datasets in order to apply the software as a guide to apply the software to your own datasets. So first of all, what we will need to do is to install MetaCluster, and this can be done very conveniently out of R directly by basically using DevTools, and here you see it's basically just a one-line or two-line install, right? So if you have DevTools installed, we can use the command install GitHub, and then it kind of depends what system you're running with respect to how the authentication works. And here we go. Okay, so this is for Mac. We use this command, and here you can see it actually lets you know if there have been any changes since the last install, and then it actually installs an update. So now we have MetaCluster installed, and now we can load it the same way we would load any other R package. Okay, next thing that we need is the data sets, and we've set a link here to the OneDrive folder where we stored all the data sets that we've been working with throughout the paper. And our gene cluster definitions come from Schlapper et al., from the Schlapper et al. 2017 paper, and actually we've been working with the Arabidopsis thaliana gene cluster predictions. And so once we've downloaded data sets, we actually, as described here, for running the software, we need to set the working directory to the folder where we placed or downloaded the data sets. Okay, so once that's done, we can actually start loading the data sets. And let's have a brief look at the parameters that the load dataset function takes. So that is actually, the first one might be of interest is the input format. I'm going to say something about that in a second. The other 6 parameters refer to the file names to the given data sets. So first of all, we have the gene cluster data set. That is the definition of our, in our case, metabolic gene clusters. And of course, you can actually input any type of gene cluster definitions. So we've been applying the system to work on metabolic gene cluster. That is enzymatic genes collocated on the genome, and they were predicted in this large-scale study by Schlapper et al. And this is our input, and then also we have pre-computed 2 differential expression matrices, one represented as full changes, the other one as p-values, and we store them as text files. And once we recreate the matrices from these text files, we have genes in rows, and the columns are differential expression samples, and that is actually our 2 text files, 2 lists basically, that we also load in order to assign them to the rows and columns of these matrices. And then there's one last file that's very important, that is the experimental annotation. And yeah, we're gonna look into these files in a second in order to get an impression of how that looks like. And basically you can take these data set as a reference as how your custom files should look like, what is essential what is needed for the algorithm to run properly. One more thing about the input formats real quick. So if you would be working with your own data sets, you would set this to custom. In our case, we have two presets here for the data sets coming from the plant cluster finder predictor by Schlaffer et al. And this is PCF217 or PCF217 enzymes only. So here, as you see, the default is set to enzymes only. So what that means is that the cluster finder allows for within a set of collocated enzymatic genes on the genome, it also allows for some non-enzymatic genes being lying in between those metabolic enzymes. And actually for our condition-specific analysis, we can actually tell the algorithm to ignore these specific, these non-enzymatic genes. So this is some special case of the PCF217. But again, if you would be working with your own data set, you would set this to custom, and then we have prepared some custom example data so that you can see how you would need to define the column names of this particular file storing your gene cluster definitions. Okay, two more things about the annotation file. So let's quickly look into the annotation files, how the experimental annotation file is set up. So what we do have, we have a series ID that is the experimental series where the differential expression data that you use comes from one particular, and of course you might have as many experimental series as you like, and also we might have different samples of differential expression, so that could be assigned to the same experimental series. So for example, given that we have a time series experiment that has samples at different time points where you compute differential expression at different time points, and along with this you assign, often of course this comes from the description of the experimental series, but as we did in the paper, we kind of summarized into a bit more high-level condition descriptions, and the algorithm allows for two conditional treatment annotations to be set. So this is why we have condition treatment 1 or 2. If you do not have a second one, you can simply leave this blank, and then also you assign the tissue, and then you, for each experimental sample, you basically set a unique ID, and that actually these unique IDs, these do link to the corresponding columns in our differential expression matrices that we've been just talking about. So actually, yeah, that's what you need to make sure, that there's actually the correct correspondence here. And for your own data sets, you would actually need to keep all the column names, but everything else is basically up to you. Okay, so yeah, this is, and then as I mentioned, if we look into this is the genes text is simply a one-column text file that stores the row names, the matrices, respectively the genes, and here we store as a simple text file that stores the column names, and that are these unique IDs that I've just been mentioning. Okay, and actually we renamed this file into sample IDs differential expression. Okay, so now the next thing that we can do in order to run the algorithm is to load our data. I prepared this already, but let's just copy paste it one more time. So we run data has been loaded. We can just quickly inspect it a little bit. Okay, so as you can see here, it actually loaded all the data files that we need, and we have our two differential expression matrices loaded. It's actually the same, kind of like two matrices of the same differential expression. Of course, it's two different ways to represent it and needed by the algorithm. Then we have our experimental annotation, basically the table that we just looked at. Then we have our gene cluster definitions here. In this case, in Schlaffer case, we also have some gene names and reaction IDs with some additional information. And then we have two additional, basically pre-processed from the experimental annotation tables. And this is actually needed by the algorithms, like a summary, a distribution actually of the individual conditions. And actually from this one, that's needed later to compute the enrichment tests, whether a certain cluster has an enrichment in a certain condition or tissue. Okay, so this is actually what comes from the load datasets function. Once we have this, we can then actually go over to execute the run metacluster function from the package, and that will actually do all the heavy lifting and compute our gene cluster definitions according to the algorithm as described in the paper. And there's a series of, as you can see here, there's a series of parameters in addition to our data sets that are at the top here, and then we have a bunch of additional parameters, some dealing with parallel processing. So this is actually kind of nice that it allows for, given a number of cores that you can spend for the algorithm, and it's actually going to spawn some processes to do computation. And then we have a series of p-value thresholds. And one thing of interest is this Boolean parameter here that actually lets you state whether you would like to load the codifferential analysis results. So this is gene by gene matrices from file, or whether you need to compute them from scratch. And that actually refers to and actually comes in handy in case that you would like to work with the same differential expression data. And if you have worked with the same differential expression data before, but now want to process new gene cluster definitions. So in our case here, we actually set this to yes, because we have been pre-processing these matrices, and you've just downloaded them along with the other data sets. They're stored in the temp folder, and since we're working with the same differential expression data, we can actually just load these matrices. If in case that you would like to change and use custom differential expression data sets, you need to set this to no in order for the algorithm to compute these, what we describe in the paper as co-differential expression matrices from scratch. And actually then you can, in case that from that point on, only your gene cluster definitions change, but you're gonna be working with the same differential expression data for quite some time in the future, you can actually set this to yes. You don't have to recompute that all the time. It's going to save you quite a lot of time, because this is actually quite some time-intensive process. Okay, so now we can, we're actually ready to run the pipeline. So let's do this, and here we go. Now that the algorithm is finished, we can actually find our results in the results folder. There's actually a folder also as a parameter name here that you can give the algorithm so it knows where to store the temp or result files. And actually the folder directory would not exist, it would just create it. Yeah, let's have a quick look into these tabular result here. It gives you quite a lot of detail for each gene cluster, what conditions, what tissues have been annotated, with what levels of significance with respect to the individual steps and individual analysis types that the algorithm computes, and then summarizes what is the final p-value of that cluster, and so on and so forth. So this might be very nice, gives you a bit more detail on the number of co-expressed genes and co-differentially expressed genes, and so on and so forth. Yeah, so that's actually also, this is stored in the results files, also given as an output of the algorithm directly in R. And then if you would like to have this in a bit more visually appealing way, you can also, we have this evaluate and store results function that takes the results as input and then actually creates some heat maps. And yeah, so we can actually go into results, and then here we have our text file, and then here we, per cluster identifier, we can actually get a little bit more nicer heat maps in order maybe for some publication or further analysis. Okay. Yeah, so that's it for our little hands-on tutorial on how to use our MetaCluster software. If you liked it, please feel free to leave a thumbs up below. And if you have any question, feel free to contact us and we'll get back to you.